Hey guys, we're going to finish up our notes over Chapter 10 concerning cell growth and division. I am going to review some things that we've already talked about as well, so let's get going. Here's a picture of a liver cell that has almost completed the process of cell division. At this point, it looks like we're about to complete cytokinesis, which is the division of the cell. And remember that during mitosis, we split into two roughly equal daughter cells that contain the same genetic information. So why do cells divide? There are certain problems that are caused if the cell does not grow and divide. The larger the cell becomes, the more demands the cell place on its DNA, and then the more trouble the cell has moving enough nutrients and waste across the cell membrane. So some factors that affect cell division: DNA overload. There's an information crisis. There's not enough information to pass through that large of a cell. The exchanging of materials, like I said, getting food into and waste out of the cell in a good amount of time that is healthy for a cell. And then the ratio of surface area to volume. Volume increases more rapidly than surface area. This is when I use the example of our high school. We only have really 2 roads that take us to the high school, which ends up causing a lot of traffic. Okay, so if you think of the roads at like the surface area in our school as the cell. If we had more roads or more surface area to get to the school, we'd be able to get here a lot faster and we wouldn't hit so much traffic. So before a cell becomes too large, what it's going to do is divide into 2 new daughter cells through a process we know as cell division. This will solve all 3 of those problems. I'm going to take you to a different window now and review the process of the cell cycle. We just answered one of the questions of why cells are going to divide, and that has to do with limits to cell growth. Other reasons is because cell division allows us to replace damaged cells as well as grow into larger organisms. Okay, so the cell cycle has 3 main steps with several sub-steps within each one: interphase, mitosis, and then cytokinesis. During interphase, the cell spends its time growing, carrying out its function, making a copy of DNA, and then preparing to divide. I'm going to break that down and show you each part. We have growth, which remember is the G1 phase. For a general cell, that might take about 9 hours. Then we have the S phase, where the DNA is copied, and then the G2 phase, where the cell is going to prepare to divide and go through the process of mitosis and cytokinesis. Okay, so mitosis is divided into 4 separate phases. We start off with prophase, metaphase, anaphase, and then telophase. So let's break down the name or the process. The definition, for mitosis is a process that ensures each cell is identical to one another. Remember, it is the division of the nucleus itself. And for this reason, each chromosome, nucleus, and mitochondria must be replicated exactly to avoid automatic cell death, which is also known as apoptosis. What would happen if a new cell did not have the same number of chromosomes as the original? Generally, like I just mentioned, the cell will be programmed to commit self-death, or something called apoptosis, to get that bad cell basically out of the organism, or not put into play. Freehand. Here's our nuclear membrane breaking down. Our chromosomes have been copied. They are attached at the centromere, the sister chromatids, which are just duplicate copies of the chromosome. And then you have your centrioles and microtubules starting to form to help move those chromosomes to opposite ends. All right, metaphase is the next phase. And in metaphase, the chromosomes line up in the middle of the cell while the spindle fibers hold them into place at the centromere. So here you have your chromosomes. You can see the spindle fibers are attached to the centromeres and are helping them line up in the center of the cell. Next is anaphase. During anaphase, the spindle fibers shorten and pull the chromosomes apart at the centromere. Now this is a close-up of an onion root tip cell that we've already looked at, so it's really hard to see the centromeres at this point, but you can see that they are being pulled away to opposite sides of the cell, and that's how I like to remember what anaphase is, because it starts with an A, it's pulling away. And this is going to help us create two sets of identically genetic information for the two future cells. Finally, we move into telophase. It's the last phase of mitosis. In telophase, the membrane of the cell begins to pinch inward, creating a cleavage furrow if you're talking about an animal cell. In a plant cell, as we have pictured here, it's called a cell plate that will form. The nuclear envelope begins to reappear at this point as well, and you can begin to see the beginnings of two separate cells. All right, lastly, we finish up the process of cell division with cytokinesis, which again, it's not a phase of mitosis, it's separate, but it is part of that entire M phase or cell division. And during cytokinesis, the cell completely divides, and you end up with two cells that are identical to the original. So you can think of, remember, cyto means cell, kinesis, to divide, so the division of the cell. And you end up, like I said, with two identical copies of the previous cell that started the cycle. Right, go ahead and watch this short video that is going to show you the overview of the cell cycle we just talked about.
And M. A fifth stage, G0, is where the cell remains indefinitely in. There are four true stages of the cell cycle, G1, S, G2, and M. A fifth stage, G0, is where the cell remains indefinitely until it begins the cell cycle again. Some cells may never leave the G0 stage. Others that go through division rapidly, such as bone marrow cells, may skip the G0 stage entirely. Cells begin the cell cycle by entering interphase. In interphase, the cell prepares for division by duplicating DNA. Interphase includes the stages G1, S, and G2. In G1, the cell synthesizes DNA and protein and increases organelle and cytoskeletal elements. The chromatin consists of long, slender rods jumbled together in the nucleus. S is the synthesis stage. Nuclear DNA replicates along its centrioles, which are small structures made up mostly of microtubules that associate with the chromosome's DNA. The chromatins begin to coil so that they shorten and thicken. In G2, RNA and protein are synthesized. Proofreading also occurs. The DNA replicated in the S phase is checked for mistakes. If mistakes are found, the cell repairs them or receives a signal to self-destruct, called apoptosis. The difference between G2 and G1 is that in G1, DNA and protein are made, while in G2, RNA and protein are made. In the M stage, the cell divides into two identical cells through divisions of the nucleus, mitosis, and cytoplasm, cytokinesis. Over 5 different stages, the centrioles pull the chromosomes apart and the cell divides in two. First, in prophase, the duplicated chromosomes appear and then split into identical halves called chromatids, attached by spindles called centromeres. The nuclear membrane disappears and spindle fibers form in the cytoplasm, radiating from the two centrioles located at opposite poles of the cell. In metaphase, the spindle fibers pull the centromeres, aligning them in the middle of the spindle or the equatorial plate of the cell. The two chromatids are clearly visible in this stage. In anaphase, the centromeres split, pulling sister chromatids to opposite sides of the cell. By the end of anaphase, 46 chromosomes lie at each side of the cell. When chromosomes are separated, each sister chromatid is considered a new chromosome. In telophase, a nuclear membrane forms around a group of 46 chromosomes. The spindle fibers disappear and the chromosomes uncoil. Cytokinesis occurs and the cytoplasm divides into equal parts. Two identical daughter cells have been formed. Each new cell moves back to the G0 stage and is ready to begin the process again.
Okay. So that was an overview of the entire cell cycle. Now I'm going to take you back to the PowerPoint. We have just a few more notes to go over. And we are almost there. Okay, so section 3 in chapter 10 talks about how we regulate the cell cycle and what controls there are on cell division. If we had cells grown in a lab, they will continue to divide until they come into contact with other cells. Then they stop growing. When you remove cells, the cells will divide again until they touch other cells. So this shows that controls on cell growth and division can be turned on and off. There are proteins that are known as cyclins that regulate the timing of the cell cycle. There are some that are internal regulators and some are external regulators. The internal regulators are proteins that monitor and respond to events inside the cell, hence the name internal. External regulators are proteins that respond to events outside of the cell. So external, outside. They can direct the cell to speed up or slow down their cell cycles as needed. So what happens when cell growth is not controlled? This is when we get things like cancer cells. They do not respond to signals that regulate the growth of most cells, and as a result, they form masses of cells known as tumors that can damage the surrounding tissues. There are many causes for cancer. A few are listed here: smoking, radiation exposure, viral infections, and sometimes it just has to do with genetics. Here is a picture of somebody with lung cancer. You can see this large mass in the upper right corner of the lung, right here. And this is obviously, it's a very large mass. Here you have a ruler kind of showing you how big it is. And you can see how it would affect you being able to breathe properly. And if this, if this continues to grow, your lungs aren't going to be able to function properly, which can lead to even more problems. So all cancers have one thing in common. The problem is the control over the cell cycle has broken down. What scientists have found, or researchers have found, is that many cells have a defect in the gene P53. This gene normally stops the cell cycle until all of the chromosomes have properly replicated. Cells lose the information they need to be able to respond to the signals that normally control cell growth when there is a defect in this gene. Okay, and that is all. Please feel free to go back and go over the video again, pause as necessary, and we will see you later.